Warm welcome to this Life Science Summit, Göran, and please go ahead with your presentation.
Thank you so much. It's a pleasure to present Cantargia here at this virtual investor meeting. And if we go directly to slide number 3, I can present a little bit more about Cantargia. So we are an antibody company. We are completely virtual at this point in time, so we have no labs. We outsource everything to professional subcontractors. Our lead asset is called CAN04, and it's in a phase 2A clinical trial, both non-small cell lung cancer and pancreatic cancer, where we study combination with the chemotherapy as, let's say, the most important part. We're also doing monotherapy. We're starting new development in solid tumors where we do combinations with immune therapy, and that start later on this year. And we also have opportunities to expand CAN04 into other cancer forms, and we hopefully can present more about that in the future. Our second product is called CAN10. It's in preclinical development right now for systemic sclerosis and myocarditis. And we have a third product called CANXX, which is, let's say, a platform product identifying new antibodies for us. What's unique about Cantargia is that all our antibodies are directed against one molecular target called IL-1RAP. But by binding different epitopes, you get very different properties of these antibodies. So if we go ahead to slide number 4, so a quick summary of Cantargia is that our lead asset then works as an immune therapy antibody. We have presented phase 2A interim data that were very positive, and obviously come back to that. But we've shown that combination CAN04 chemotherapy gives a much better, higher response rate than what is expected from just chemotherapy alone. IL-1RAP, our molecular target, is found on a large number of tumor forms and also on hematological cancers. So there are plenty of opportunities to expand this program. But in short term, we'd like to stay focused on pancreatic cancer and non-small cell lung cancer. There is also an opportunity, since this is an inflammatory pathway, to work in autoimmune inflammatory diseases, and that's where we're starting out in systemic sclerosis and myocarditis. We also have a very robust patent portfolio. We have global patents on not only our compounds, but also on IL-1RAP as an antibody target for oncology therapy until 2032, which means that we actually have a monopoly situation here, which is obviously something we would like to explore as much as we can. We have a market cap of around $190 million US. We have a very strong cash position. We recently, or in February, raised 410 million Swedish, and already had 150 on the bank end of Q4. We're presenting the, let's say, the updated results in our quarterly report tomorrow, so I'll not go into that, but I don't think you should expect any major surprises. We have a very strong shareholder list with strong institutional investors backing us, with AP4 being the biggest investor. We have Swedbank Robur, Alecta, First AP Fund also being major shareholders. We have some specialist investors like Danish Sunstone having 6% of the company, and under Morgan Stanley you would find HBM, a Swiss specialist investment fund, and Handelsbanken Funds also have some specialist competence on board. So if we go to the next slide, we can just say that the market we are addressing in, if we just look at pancreatic cancer and non-small cell lung cancer, it's huge. Together there are about 2.5 million people being diagnosed with these two diseases annually each year. Most of these patients die very quickly, so the mortality rate is well above 2 million cases per year in these diseases. So, and if you then start to think about what could be the next steps here, it's just an enormous potential for new patients with our therapy. So if we then focus a little bit more on our lead antibody CAN04 and jump into slide 7, we have got some very strong support for what we're doing. And in 2017, almost at exactly the same time point as we started our phase 1 clinical trials, Novartis, who has, let's say, one type of antibody against the same molecular pathway, presented very interesting data on an antibody against IL-1 beta. They did their trial in more than 10,000 patients who had previous cardiovascular or myocardial infarction, but they saw a very strong signal in reduction of lung cancer. And obviously myocardial infarction and lung cancer are linked because of smoking. So actually they reduced the risk of getting lung cancer by 67% in this patient population, and they reduced the risk of death by 77%. And what Novartis has done is that they've taken their antibody into phase 3 in non-small cell lung cancer and dropped the cardiovascular opportunity, and are having one trial in, let's say, early stage lung cancer, one in first line metastatic and one in second line metastatic. And at least the two latter trials will probably read out late this year or next year. We don't really know that, but that's what the communication says. So something to follow up very with great interest from us and from investors. So if we go to the next slide, we can just see how does Cantargia's antibody differ from what's in development. So the interleukin-1 system, which is what we are attacking, consists of two different cytokines called IL-1 alpha and IL-1 beta. And Cantargia targets the receptor for these two through IL-1 RAP, which means that we block the activity of both IL-1 alpha and IL-1 beta. And we also have engineered our antibody to stimulate the immune system to attack cancer cells. So it means that we have a very strong IL-1 alpha blocking activity with two green pluses. We have a very strong inhibition of IL-1 beta with two green pluses. And we have ABGSC, which is stimulation of the immune system. If we, for instance, compare with Novartis, they have an antibody which is just binding IL-1 beta and removing it from the blood, which means that they got very potent effects on that, but still they are not having a complete solution to blocking IL-1 in general, and they have no activity against the tumor cells. So we follow what we're doing with great interest, but we see that we have several advantages compared to their concept. And there are also other, like ex-biotech Janssen's antibody, which is blocking IL-1 alpha, which is being developed for dermatological autoimmune diseases, but again, just blocking one of the two cytokines and not both. So then we can go to the next slide. So in late 2019, we published our first phase 2 combination data, and we show that in both at an early stage with limited number of patients, and I have to be very frank about that, we saw that we got more than 50% response rates in both pancreatic cancer and non-small cell lung cancer. This was obviously much higher than was expected from historical data with chemotherapy alone, where you have an expectation of somewhere around 20-25% with chemotherapy alone. And in non-small cell lung cancer, we also saw a complete response, which means that all the tumors were gone after therapy. And if we go to the next slide, we can see that, look into the patients a little bit more in detail. So if you have the RNC patients in non-small cell lung cancer, you can look at, for instance, patient N1, which is the patient getting the complete response. That patient had the primary tumor left in the lung, but also had other lung metastases. And after 2 months of therapy, all these tumors were gone, and after 4 months of therapy, were still gone. So that qualified for a confirmed complete response. And then you can also look at, for instance, patients P2 and P4 in pancreatic cancer, where you have two patients with a very good partial response. P2 had a strong tumor reduction already of 2 months, and that tumor reduction has been, let's say, sustained up to 6 months when these data were reported in December. P4 is a similar story. Also, the CA19-9 levels, which is a biomarker for pancreatic cancer, so you could say it's a PSA value of pancreatic cancer, was reduced by 90% in these two patients. And also in a patient called P3, which had a so-called pseudo-progression, which is sometimes something you can observe with immune therapy. So again, suggesting that we have very interesting results here. And obviously we are now recruiting more patients, and we are getting more and more data, and we hope to present them later on this year. So if we go to the next slide, slide 11, we can see how the trial is designed. So the trial was a combined phase 1, phase 2 trial. Phase 1 data was presented at ASCO 2019 in an oral session, and I think we got all the answers we wanted from that trial. We showed that our drug is very safe, and we also got a very pronounced effect on some relevant biomarkers showing that the antibody is really doing what it's supposed to do. It's reducing inflammation, reducing interleukin-6 and CRP, for instance. In phase 2, we have continued with monotherapy. We have done a difference here is that we've taken biopsies from all patients before and during therapy. We had hoped to present all these results by now, but due to the COVID situation, the lab performing these tests had to close down temporarily, and once we're opening up, we will get the complete data set hopefully soon and present that. It's going to be very, very interesting to see what's happening in the tumor when we do our therapy. But in the combination arms, which is really what we are more from a commercial point of view interested in, is that we have recruitment ongoing both non-small cell lung cancer and pancreatic cancer. It's ongoing despite the COVID situation, and there's lots of interest from patients to enter. We expect the last patient to initiate the therapy in Q3 for pancreatic and in Q4 for non-small cell lung cancer. And altogether it will be 31 patients with pancreatic and 31 patients with non-small cell lung cancer. And based on the data we get there, we will obviously communicate more about what is the next step and how will these different indications be expanded, and perhaps also looking more into subgroups where we really see that patients benefit. And we're also starting a new complementary trial in the US later on this year, and that will be in combination with immune therapy. So if we go to the next slide, slide 12, it's just showing a little bit of what we're trying to achieve when we combine with chemotherapy. So chemotherapy is in one way a very effective anti-cancer treatment, but its problem is that it's very toxic and the effects are quite short-term. And one reason for short-term effects is that we build up some kind of chemo resistance very quickly. And what we've shown in mouse studies, and it's also been shown by other groups, is that certain chemotherapies induce resistance by upregulating the interleukin-1 system, which is exactly what we are attacking. So what we see in mouse models is that once we attack interleukin-1 together with chemotherapy seem to work much longer and much better. So we're entering a situation with 1 + 1 = 3, and that's exactly what we're hoping to see in patients as well. So we can skip slide 13 and go directly into slide 15. And this is just showing that IL-1RAP, which is our molecular target, is not only expressed in non-small cell lung cancer and pancreatic cancer, but in those diseases, approximately 70 to 80% of patients seem to overexpress the antigen on their tumor tissue, and in a tumor microenvironment, all patients overexpress that. But there are lots of opportunities in other solid tumors, and especially in head and neck cancer and bladder cancer. We know that it's very high overexpression. It's cancers that are treated with similar strategies as non-small cell lung cancer, and we think that we can expand very quickly into there. And that's then taking us into slide 16, which is the trial we're starting on or preparing right now, and we hope to get the first patients in during Q3 this year. It's a study which will be done in the US only. We do a combination with the PD-1 therapy with Keytruda in patients that do not longer respond to this type of therapy. The IND was granted recently by the FDA, and we hope to open up the first site as quickly as possible, having in mind that there are obviously not a straightforward road forward as you would hope due to the COVID situation. But the indications we are looking into would be non-small cell lung cancer, head and neck cancer, bladder cancer, and malignant melanoma. And we're working with very good, strong US centers, and University of Pennsylvania will be the lead site with Professor Roger Cohen as the lead investigator. So that was really what we're doing in cancer. So then we can go to slide 18 and just very briefly mention what we're trying to achieve in CAN10 in systemic sclerosis and myocarditis. So these are diseases driven by 3 different cytokines, IL-1, IL-33, and IL-36, together with several other mechanisms, of course. But the interesting thing is that IL-1RAP is contributing to the effects of IL-1, IL-33, and IL-36. And CAN10 is an antibody blocking all these 3 cytokines at the same time, which means that we can attack the disease in several different steps in the disease development. So we're super excited about this project and how we can move it forward. So if we just look at slide 19, these are some initial experiments in an inflammatory model where we can see that our antibody CAN10, which is black here, has an effect which on the inflammatory response, which is much better than just blocking interleukin-1 beta, which would be the yellow. It is not as good as chemotherapy, but chemotherapy is really killing the immune response in this model, so we didn't expect that. And the idea is really to have something which is much safer than chemotherapy. So if we then go to slide 21, this is really the news flow we're expecting in 2020. So we are presenting new preclinical data on chemotherapy combinations, which is really, let's say, the cornerstone of what we're doing in the clinical phase 2 right now. So more preclinical data around that will be presented at AACR, which is in June 22nd, 24th. We are starting out our Keytruda combination trial in the US Q3. We expect to get the phase 2A combination results in pancreatic and non-small cell lung cancer later on this year. Once our lab opens up to do the biomarker and biopsy results, we will do much more on that as well and start to present that to the market. And obviously we will communicate how we will take the next steps with combination therapy with chemo. And we also so we like to stay focused in pancreatic and non-small cell lung cancer. On the other hand, we also like to explore more opportunities. So we are going to broaden our case and look into clinical trials outside CAN4, and that will also be communicated later on this year. And CAN10 is in preclinical, but we are obviously doing lots to do progress here. And one of the bottlenecks would be the production development, and from a timeline perspective, and that will also be communicated as we progress. So if we then go to slide 22, we have a summary of the company. And I think we can just have that as a background while you were asking questions to me. So thank you very much for your attention.
Thank you for that, Göran. A lot of interesting news flows indeed coming from you here recently. Starting with your recent press release about the preclinical data and CAN04 in combination with chemotherapy. Could you perhaps, spend a few minutes here and give some more granularity and what you are most satisfied with from the outcome? What does it mean for Cantargia to reach this milestone?
Yeah, so obviously there is, when it comes to chemotherapy in general, there is, let's say, lots of enthusiasm around immune therapy. On the other hand, most people, or actually much more people than are treated with chemotherapy than with immune therapy, and it's going to be like that for a long time to come. So let's say understanding how our drug can be combined with chemotherapy isn't really it's not a niche, it's really a big area where the medical need is huge. And let's say understand this from a preclinical point of view, so we know exactly how we should position CAN04 in combination with chemotherapy in the clinic is of great interest. So you should really see this as we're doing clinical activities, we're doing preclinical activities, and we try to make these activities go hand in hand and progress forward.
Thanks.
So then I think when it comes to the actual results, we need to wait until AACR before I can really comment on that. But the expectation should be is that it's more granularity around the mechanism of action and why we believe CAN04 can increase the activity of chemotherapy.
Yeah, gotcha. Thank you for that. And please, if you could elaborate a bit more on your IND application that was approved by the FDA here in early May as well. What did this mean for you and the study that you now will conduct with what patients will be included and so on. Talk about the awareness about this and the US market in general for you.
Yeah, no, so the US market is obviously it's the biggest market for pharmaceuticals, and obviously it's going to be the biggest market for CAN04 if we go all the way through. And FDA is also, let's say, the most influential regulatory authority. So starting clinical activities in the US will lead to an increased awareness of our drug and starting clinical. So now we have one leg where we do chemotherapy combinations, but it's obviously that we can see synergies with immune therapy, and starting those activities in the US is of great importance. But there is also a third aspect of getting the IND in place, because with the IND in place, we can start to, let's say, discuss several regulatory designations with the FDA, and we don't have enough data yet, but if data continue to look good, obviously we can submit for breakthrough designations or fast tracks and all these as well. So, but we need to have the IND. So the IND was really the door opener to have those discussions.
Yeah, Göran, I think that time flies here. It's been good presentation here. Thank you very much for that. Stay safe and thank you very much for joining this meeting.
You too. Thanks a lot.